We are now starting our last unit, which is going to be on the muscles. And so over the next couple weeks, we're going to go over all different types of topics that have to do with muscles. Today, we're going to talk mainly about the anatomy of muscles, kind of the cells, and next time we'll start getting into how muscle contractions actually take place, which is going to require a lot of information kind of to do with the nervous system. So muscles are going to comprise nearly half of your body mass. And muscles are going to be able to take kind of ATP and transform it into actual work. And so we can go through the process of cellular respiration and all that type of stuff where we go from glucose to ATP. But now muscles are going to be one of the kind of the structures that's going to use that ATP directly to do work. So it can be, walking, lifting things, your heart beating, things like that. So ATP is going to be a very important component of the process of allowing muscles to contract. Now, we're going to look at today types of muscle tissue. So we're going to talk about the 3 different types that we've already gone over before, but we're going to go a little more in detail about skeletal, cardiac, and smooth muscle. We're going to talk about the characteristics of muscle tissue. So what are the functions? What are some of the different properties of muscles? And then we'll ultimately go through some of the muscle functions. Okay, we're mainly going to focus today on the types of muscle tissue, the actual components of muscle, so the cell going down to the cellular level and the myofilament level, and then later on we'll talk about different muscle functions. So you're going to hear a lot of different words that start with either myo, mice, or sarco, and all of these prefixes have to do with muscle. So we're going to talk about, for example, myofilaments. That's going to be the muscle filaments, myofibrils, things like that. So make sure that when you read these terms, you are automatically starting to think muscle. So, for example, the sarcoplasm is going to be the cytoplasm of a muscle cell. And we're going to talk about the sarcoplasmic reticulum, which is the endoplasmic reticulum for a muscle cell. So it has a different name. Now, there are 3 main types of muscle tissue. We have skeletal, cardiac, and smooth muscle. And so these are going to be the 3 that you need to be able to look at under a microscope. Well, basically histology, since we don't have microscopes, but look at images of these different skeletal, cardiac, and smooth tissue and be able to identify which is which, okay? And they do have quite different appearances, so hopefully that won't be too difficult, but we'll talk about kind of what are the things you need to look at when you're trying to identify these different types of muscles. You're going to hear the term muscle fiber, and that actual muscle fiber is going to be kind of only used when we're referring to skeletal and smooth muscles. Okay, and so these are going to be cells that are elongated. When we're talking about cardiac, it's going to be slightly different. So the first type is going to be skeletal muscle. And this probably is the one that most people are familiar with because if you like to weightlift or exercise, you use these skeletal muscles all the time. These are going to be the ones that are going to allow for all of our locomotion. They are going to be packaged into kind of what we would call organs. So an actual muscle would be an organ that's attached both to bones and skin. Okay, and so we'll talk about the different components that are going to make up that organ. The skeletal muscles are going to be the longest fibers of the body, and they're going to have these striations. The word striation is going to come up many times, and it just means stripes. So if you look at the picture over here, over here, you're going to see that there are lots of stripes on the actual muscle fibers. These muscles are going to be under voluntary control, which means that we are consciously thinking about moving them. I mean, yes, there are some times where you have spasms of your muscles where you're not thinking about moving your muscle, but most of the time we are consciously deciding to, like for right now, I'm speaking to you, which is using a lot of different muscles in my face. That is voluntary. And so I can think about it and I can do it. I can get up and go walk somewhere. Those are under voluntary control. These types of muscles are going to be able to contract rapidly, but they also will tire easily. So at one point we were going to talk about different types of skeletal muscles where we have fast twitch and slow twitch muscles. Kind of depending on the proportion of each type, some of them are going to be able to tire, or they tire faster than other types, but we'll get into that detail a bit later. But compared to the other type of muscles, these ones are going to contract very rapidly, they tire easily, but they're very powerful. And when you're thinking of skeletal muscle, think things like, obviously the word skeletal, striated, and voluntary. So those are going to be keywords. Next group is going to be the cardiac muscle, and this is going to be only found in one location, in the heart. And so cardiac hopefully is a term that you associate with the heart already. So this is the only place you're going to find it. It's going to make up the majority of the walls of the heart. And it also is going to be striated. So if you kind of look in this image right here, you can see the striations. It looks a little bit different from skeletal muscle, and hopefully you can kind of see how it's branching and not so much parallel lines, fibers as we saw previously. These type of muscles are going to be autorhythmic, which means that they have a rhythm to them, and it's something that is not consciously thought about. So we don't control our heartbeat. We're going to have like pacemaker cells that are going to be in charge of that. It's controlled by the nervous system. So it's involuntary, cannot be controlled consciously. I mean, you can definitely scare yourself probably consciously and then increase your heart rate, but it really is not under our control in a conscious way. Okay, so we won't talk much about the cardiac cycle or anything with that in this portion of the class, but your heart is going to contract at a steady rate due to the heart's own pacemaker. So there are cells that are going to be in charge of kind of sending out this signal and keep the heart contracting. People that have issues with their heart sometimes have pacemakers put in, which is going to then control the heartbeat for them. Okay, but the nervous system is going to be able to increase and decrease the heart rate depending on activities and other types of things. So keywords for the cardiac muscle are going to be obviously cardiac, striated, and involuntary. For smooth muscle, this last type, this is going to be kind of found in the walls of many of the hollow organs, including the stomach, urinary bladder, and the airwaves. They're also going to be in kind of large arteries, in the eyes. There's lots of places that you can find this. Some of the functions are going to be propelling urine. The ones in the digestive system are going to mix food and also kind of move food from one place to another. They're usually going to be in charge for dilating and constricting your pupils and regulating blood flow. They are not striated. So if you look at the image, you can see the cells, you can see the nuclei, and you can also see that there are no distinct lines or striations, stripes. These are going to be involuntary as well, so just think about it. You are not consciously thinking about moving the food from your stomach to your small intestine. That's just happening. And so these things are going to be under involuntary control. Okay, so they're going to be controlled by the endocrine and the autonomic nervous system. So for this, the keywords would be visceral, because like a lot of them are going to be part of the visceral organs. They're non-striated, and they're going to be involuntary. Here is a comparison of the muscle types. In your lab, there is going to be a table where you need to fill out kind of the different appearance, location, cell types, number of nuclei. And so here is a grid diagram that kind of puts all of that out, and it's all in one place. Okay, so for example, if we look at the nucleus, for skeletal muscle, you're going to notice that there are a lot of nuclei, and so they're multiple nuclei, and they're on the periphery of the actual muscle cells. For smooth muscle, they are going to tend to have one nuclei per cell, and it's not peripherally, it's not kind of on the edge, it's actually just going to be a central location. And then for the heart, the cardiac muscle, these are also going to have one centrally located nuclei. So that's another way to identify these, is looking at where are the nuclei. So here's some characteristics of muscle tissue. So all muscles are going to share kind of the four main characteristics. They are going to be excitable, which means that they are going to be responding to the nervous system and will be able to receive and then kind of respond to a stimuli. We will get into the details of how this actually takes place with the motor neurons that are going to cause muscle contractions. They're also going to be contractility. So these cells are going to be able to contract, which means to shorten. So when we have contractibility, just think about when you're flexing your arm, you are shortening the length of some of those muscles and contracting them. Extensibility, so muscles are going to be able to be stretched. So if you see in this diagram right here, kind of hard to see, let me maybe use white. Right here, these muscles are being stretched, so not only can they be contracted, but they can be extended. And so we'll talk about different motions of the muscles, and extensibility is going to be one of them. And elasticity, so our muscles are going to be able to kind of recoil to resting length. So yes, they can change shape and length, but they are going to be able to go back to their normal kind of resting length. So here are a couple of different functions for muscles. So they are going to be allowing us for locomotion, so moving, manipulating things. We're not like plants where we can't move our bodies and kind of walk away. We are going to be able to move through our environment very easily and manipulate our environment, and that's all going to be due to muscles. Muscles are also going to help us maintain posture and body position. So if you're sitting in a chair or standing up, the reason why you're able to do that and not flop on the ground like a kind of a slug is because we have muscles that are allowing us to stand erect. They're going to help stabilize joints. So we talked about joints very recently. So muscles are going to be really important in the stability of those joints. Trying to keep, if you have joints that are going to be really important in movement, they're going to need to be stabilized by muscles. Muscles are also going to be able to generate heat as they contract. And so when we kind of are exerting ourselves and contracting muscles, we have heat, in which will kind of maintain part of our homeostatic thermoregulation. Respiration is going to be very important. We have to contract and relax our diaphragm in order for us to allow oxygen to enter and CO2 to exit our lungs. Communication, so communication uses both the respiratory system and the muscular system. So we are able to shape our mouths, move our tongue, and things like that in order to produce words and communicate. And also nonverbal communication. So the way that our faces are going to display different emotions is a type of communication, and that all has to do with how you're moving your face muscles. There's going to be constriction of the organs and vessels. So we talked about smooth muscles. They're going to be important in different types of kind of functions in that. And contraction of the heart. So we're able to pump blood from one location all the way to every part of our body because of this muscular function. So a little bit more about skeletal muscle anatomy. So again, if we're looking at kind of a full muscle, it's thought of as an organ because it's going to have different tissues. It's going to have connective tissue. It's going to have nerves and blood, and it's also going to have some attachment sites. Okay, so it's going to have a lot of different things coming together to work for some type of function. Each muscle will have its own nerve, artery, and veins. You have to have lots of arteries and veins to the muscles, because having oxygen to be able to break down glucose into ATP is essential. So we have to be able to do cellular respiration, and also need to be able to get rid of kind of waste products like CO2 when you're doing this. And muscles need to be able to receive nerve impulses so that we can actually move our body. Okay, we can consciously control, again, skeletal muscle, and so it has nerve supply that control the activity. Okay, contracting muscles are going to, again, require a lot of oxygen and nutrients in order for them to be able to perform the functions that they need to perform. Okay, and need to be able to remove waste products. So here is kind of an overall picture of skeletal muscle, and we're going to now start focusing on the different components of the muscle. So each muscle is going to have a surrounding connective tissue, and there are actually 3 types of connective tissues that are going to surround the different levels of the muscle. These are going to be support cells, which kind of keep the shape and everything together, but they're also going to help reinforce kind of structure. The sheaths are going to go from the external to the internal, and so we'll talk about the external-most to the internal-most layers of this tissue. Okay, so epimysium is going to be this most external type of connective tissue. And again, remember, we've talked about lots of different connective tissues, but epimysium, so we have that mys, which means muscle, is going to be a dense, irregular connective tissue, and this is going to surround the entire muscle. So this is kind of gross, but when you think about when you eat kind of chickens or beef or anything like that, you're often going to have kind of the skin, but then under it you're going to have this connective tissue that can kind of blend with the fascia that's going to surround all of this. And so you can kind of see that layer when you're eating the food. The lower levels, so kind of the next level in, is going to be paramecium, and so this is going to be the kind of next layer. And so the epimysium is going to be, let me change my colors, sorry. So epimysium is right here, so it's on the outside. And then if we look at kind of what we're going to call a fascicle, so this is kind of one area right here. This is going to be part of the muscle. The fascicle is going to be surrounded by the paramecium. Okay, so epimysium kind of surrounds the entire muscle organ, and then we have paramecium, which is going to be around this thing called the fascicle. Once we get to the actual muscle fiber, and the muscle fiber is going to be the actual muscle cell, so the word fiber is synonymous with cell in this case, so the muscle fiber is the muscle cell. It is going to be surrounded by endomysium. Okay, so endomysium is going to be a fine areolar connective tissue surrounding each of the muscle fibers. Okay, so kind of from epimysium being around the entire muscle, paramecium around what we're going to call fascicles, which are groups of muscle fibers, into the next state, which is going to be the endomysium, which is going to be around an individual muscle cell. Now, the way that you have attachments with kind of muscle to bone is there's a couple different ways that this can happen. And one of the big things you're going to have to learn in lab are kind of the attachments in the 2 places that we're going to call the insertion, which is the kind of where the muscle attaches to the movable bone, and the origin, which is where the muscle attaches to an immovable or less movable bone. Okay, so we'll talk about for each muscle, or for many of the muscles, their origin and insertion. And so origins tend to be kind of more proximal, not always, but they tend to be closer to the body, and then their insertions would be kind of a more distal location. But again, those things can sometimes be different. Now, there are 2 different types of attachments that can be direct or indirect. And so if you have a direct, we call fleshy, this is when the epimysium is going to fuse with the actual bone and the periosteum of the bone, or perichondrium if we're talking about a cartilage connection. There is another type which is called an indirect, which is when you have connective tissue wrappings that will kind of go beyond the muscle. So the muscle is kind of ended and continues through this connective tissue. And it could be something like a tendon or something called an aponeurosis. And so in this image, we have a tendon right here. So you have muscle, the muscle ends right in this location, and then the tendon continues on. So this is going to be an indirect type of connection. And then here is one of big aponeurosis, this white portion in between. This is in the abdomen. And so you can have kind of this connective tissue that is going to attach to the different muscles. All right, so just a little bit about the structural organization of muscles at different levels. So we've talked about kind of the muscle as an organ right here. So if we have the whole muscle, it's going to be surrounded by the epimysium. The next layer down, so kind of a whole muscle organ, is then the fascicle. And so here is the fascicle. So if we go down to this level, we have now a fascicle. Fascicle is going to be a bundle of muscle cells. And so when you look at all of these little individual, you have little individual muscle fibers, you're going to see that we have multiple different muscle fibers within that fascicle. Okay, and that's going to all be surrounded by perimysium. If we then look at the muscle fiber, this is our next level, we're going to see kind of the endomysium on the outside. There's a layer right here. There is then going to be, this is now what is going to be our cell. So this is a muscle cell. We're going to have the sarcolemma, which I'm going to explain, kind of define what that is. But the sarcolemma is basically going to be the cell membrane of the muscle fiber. And then inside, which is kind of where we're going next, are going to be a bunch of myofibrils. And we will talk about the myofibrils in a second. Okay. Here's another image of that. Again, so we have epimysium, and there's a deep fascia that is around that. If we come in, we have, these are going to be different fascicles. Here's a fascicle, here's a fascicle, here's a fascicle. This is then a fascicle that we're looking at. It's going to be surrounded by kind of the perimysium, and then within the fascicle, we're going to have individual muscle fibers. So these are going to be the cells. And so then we're going to start focusing down onto an individual cell. These are long, so these muscle cells are very long in skeletal muscle. So they can go down the length of the muscle, and we're going to talk a little bit about that, okay? So now we're going to start talking about microanatomy. So we are now going to be at the muscle cell or the muscle fiber kind of level. So skeletal muscles, again, are very long and they are cylindrical. So, they're going to look like this. And we're going to start talking about the myofibrils, which are going to be running the length of the muscle. So they're going to run this direction. Okay. So we'll start using some new terms. And remember, I told you sarco means muscle. So the sarcolemma is going to be the plasma membrane of the muscle cell, okay? This right here, so this kind of outer, it looks whitish, is going to be the sarcolemma. Oops. Sorry about this, how it looks on there. I'm trying to use a new pen. So the sarcolemma, this term needs to become synonymous with cell membrane for muscle cells. Okay, so I don't want you to start freaking out when you're like, what is a sarcolemma? It is just the plasma membrane. Okay, the sarcoplasm is going to be the cytoplasm of a muscle fiber. And so it's kind of more difficult to see because it's, what we're looking at right here is it's full, there's all these fibers that are inside of the muscle cell, but any space in between is going to have cytoplasm. So it's going to be the fluid inside of the cell. Okay, and you're going to notice we have like mitochondria, so they're there, and there are going to be other organelles. Now, in the muscle fibers, you're going to have these things called glycosomes, which are going to store glycogen. Okay, so glycogen is going to be the storage of glucose. So glycogen is kind of long chains of glucose. This is so that you can store glucose for longer periods of time. So the glycogen will be stored within those. You can't see the glycosomes in this image, but they'll be there. There also is going to be myoglobin, which is going to be a protein that's going to be able to store oxygen. So one of the big things about muscle fibers is they have to be able to go kind of get started right away, be able to contract and relax, and that takes a lot of energy. And so you have to have backup storage for both the fuel, which is going to be the glycogen ultimately, and then oxygen that's going to allow for the process of cellular respiration to take place. There are going to be other modified organelles in here. The most important one that we're going to talk about is going to be actually there's a couple of myofibrils. So we're going to start talking in detail about the myofibrils. The myofibrils are actually going to be within these, if you look at these are going to be the sarcomeres. We're going to have different types of myofibrils. They're represented by the little blue and red dots, and you can also see them kind of here. So the red line and the blue lines, those are going to be myofibrils. The sarcoplasmic reticulum is going to be an endoplasmic reticulum for muscle cells. It's specifically a smooth, so this is smooth. So it's not the rough ER. So smooth ER would be what we would call it in most cells, but we're going to call it the sarcoplasmic reticulum. This is going to store calcium for us. Okay, and then we're going to have this other structure called the T-tubule. So the T-tubule is part of the sarcolemma, and it's actually going to invade within the myofibrils and between what we're going to call these sarcomeres. It's going to be really important in how action potentials are taken to kind of the actual cells or the fibers. So contraction can take place. And we're going to go over detail of all of those. Okay, so now we're back to myofibrils. So myofibrils are going to be these rod-like elements. They're very densely packed. And the myofibrils are the reason why you can see these different striations. So you can see here we have kind of a darker, lighter pattern. Within one muscle, you're going to have, one muscle fiber, you can have thousands of these myofibrils. Okay, so again, the myofibril is going to be one of these little rod-shaped structures. They're going to account for about 80% of the muscle cell volume. So if you look at this cell, it is packed full of these muscle fibers. Now the myofibril features, they have the striations, so they are the cause of the striations, and we'll talk about how that pattern starts to arise. We're going to talk about sarcomeres, which are going to be the actual kind of contracting unit on the myofibrils. And we'll talk about the myofilaments, which are going to be the actual contracting structures. Okay, so the myofibrils have myofilaments, and we'll talk about those two major myofilaments, which are going to be actin and myosin. And we're also going to talk about some more of the molecular composition of the myofilaments, because we can get down to the protein level and talk about, are they globulins, are they fiber-like, those types of things. So just as a kind of a brief aside, I wanted to mention this. So we talked about how as much of 80% of the internal muscle structure is going to be these myofibrils. And so myofibrils are going to be the contracting units of the muscle fiber. And the more myofibrils you have, the more contracting force. So when you're trying to produce stronger muscles, and so we have kind of what we would say like an active person that maybe does some weightlifting, and then this person is an inactive person, doesn't do really any weightlifting. Their muscles look quite different. And what happens is when you have exercise, your muscles are larger, not because you're increasing the number of myofibers, but you're stimulating the growth of the myofibrils within each muscle fiber. So again, so if we have kind of our muscle fiber, so this is going to be kind of that rod, the myofibrils will be the things in between here that are kind of going and going to be the contracting units. There's going to be more of those in this person in each muscle fiber than you would have in an inactive person. Okay, so let's talk a little bit about the striations, because this is one of the most interesting things about muscles, and they have these very interesting patterns and lines that are present when you're looking at the muscles. So the actual stripes are going to be formed by differing myofiber types. So we're going to have what we call kind of a dark and light band, and those are going to be part of the contracting unit. Okay, there's something called the A band. It's not A band, it's like the letter A band, which is going to be the dark region. So when you look on here, the dark regions are going to be the A band. So this is an A. And then you have next to it a light band. So there's lighter ones. Those are going to be I bands. Now within the A band, and you can't really see this very well in this image, but there's something called the H zone, which is a little bit lighter, and it's in the middle of the dark A band. And I'll show you an image where you can see this better. There also is going to be something called an M line, which is going to be a line of proteins, and they're called myomasin, that are going to kind of bisect these H zones vertically. And again, I'm going to show you what this looks like. The I bands are going to be the lighter regions, and so you can have kind of the lighter regions in between the very dark. Those are going to be the I band. Okay, and they're going to interact with the A band. With the I band, they are going to be connected to these things called Z disc or lines that are going to be proteins on the midline of the I band. Okay, and right now I assume this sounds like nonsense to everybody, but we'll start looking at images where you can kind of see these different zones. Okay, so here is what we would call a sarcomere, and a sarcomere is going to be kind of this area from I band with an A band in between to an I band. Okay, so the sarcomere is going to be the smallest contractile unit, so it's the functional unit of a muscle fiber. So if we go any smaller, we're no longer in it, it's not going to be able to contract on it, as a unit. All of these can be incorporated into a larger contractile unit, but this is going to be the smallest. We have the A band. So the A band is going to be from here to here. Okay, it's going to be where this dark red line is going to be, and then you're going to have kind of right here is going to be the I band and I band. So we have I, A, I. Okay, so that's going to be kind of what's inside of a sarcomere. I'm probably going to overlap my writing in a second. Each of those is going to have a Z disc. So this is the Z disc right here. So in between I bands, you have the Z disc. Okay, and the individual sarcomeres are just basically lined up one next to another. Okay, and so if I wanted to kind of show this in a different color, so we could say like this one's not showing a whole one, but there would be a sarcomere here, here's a sarcomere, and then another one. This is part of a sarcomere, and they're just lined up one after the other on this. Okay, so I kind of wanted to go through this. I left it blank so that you guys can practice labeling, and I'll give you more of these. But main thing again I want to just show, so we have, this is the sarcomere. Okay. Sarcomere. Okay, so that's the sarcomere. we're going to have kind of the red area from here to here. So this is going to be our A line, our A band, sorry. Now, in the A band, we talked about both the M line and the H zone. So the M line is going to be, let's see if what color will show up. The M line is kind of this area right here, these lines. This is M. Okay, and then the H zone is kind of this area right here. So the M is inside of the H. Okay, over here on the kind of blue area, we have our I bands. Sorry, I kind of drew those in the wrong spot. Oops. Let me just, okay, so here is our, and then our light area is going to be right here. So these are going to be the I bands. Okay, and the Z disc is going to be this structure right here. Those are Z. All right, so we're now going to start talking about what are these different lines, the red, blue, what are those lines actually? So for the myofibrils, we are going to have kind of an orderly arrangement of what we call actin and myosin, and these are going to be myofilaments. So again, myofibril is kind of a bigger term, and a myofilament is a type of microfibril. Okay, so the actin myofilaments are going to be what we call the thin filaments. So they are going to be the thinner ones that we have. Okay, so in this image, the blue lines are going to be the actin microfilaments. They are going to extend across kind of the I band. So they're going to be, so if this is the I band and this is the M band, or A band, so this is A, this is I, you're going to see that the blue is going to kind of occupy both of those regions. So it occupies kind of part of the I band and part way of the A band. They are going to be anchored, these blue are going to be anchored to the actual Z disc, and they are then going to interact with the next type of myofilament, which is going to be myosin. So myosin filaments are going to be these ones in red. Okay, so these are going to be myosin. Myosin are considered thick, and hopefully this is, you can see this in this image. They are actually thicker filaments than you have for the actin. Okay, so actin is going to be thin, myosin is going to be thick. They are going to extend kind of where the length of the A band. So the A band is going to be right here, and they're going to be in the entire area. So here's our A band, and that is going to be where our myosin filaments are going to be. They're going to connect in the middle with, They're going to connect in the middle at this M line. And the M line is going to then allow for them to connect. So you can see that they have to be connected to something, otherwise these thin and thick filaments would just be kind of moving around and floating inside the cell. Everything is very structured and attached to something that gives it kind of the structure so it doesn't actually move. So when you look at sarcomeres in cross-section, which are going to be these things right down here, you can kind of see different kind of images. So this one shows kind of I band filaments only, and then this next one is the H zone, and then we have the M line, and then finally this one is thick and thin overlap. Okay, so I won't ask you to look at these in this direction. You'd more need to be aware of kind of this upper structure and where everything is in relation to each other. Okay, so a little bit more about each of the different types of myofilaments. So if we look at the actual composition of these, the thick filaments are going to have this protein called myosin, and the myosin is going to have 2 chains that are what are considered heavy, and then 4 light polypeptide chains. So these are proteins, okay? The actual structures, the heavy chains are going to intertwine to form a myosin tail. And so here is the myosin tail right here. And then the thin are going to form the head. Okay, so we have all of both the heavy and the light chains for the myosin. The myosin heads are globular, so you can kind of see how they're not like these long fiber types, a little bit different. The myosin heads are going to be what actually interact with the actin, and what I want you to kind of look at is this thing that says ATP binding site. That is going to be important for kind of muscle contraction, and also this actin binding site. So this is where the actual protein, the myosin head, is going to bind to the actin. During contraction, these parts, the actin binding sites, will bind to the actin and form what we call a cross bridge, because they'll be actually connected to each other. Okay. The myosin heads are offset from each other, so if you can see, they're not all kind of at the same location. They're all over, and this is going to be important. The myosin heads are not going to all bind at once to the actin. They kind of do it at different periods of time during a contraction event, which makes sure that, you know, if all of them were released or not at the same time, that would cause some issues, but they are going to kind of work together for them to be able to form this contraction unit. So the myofilament actin is going to be thin, and they are going to be composed of these fibrous subunits. So these are going to be the actin subunits, kind of look like blueberries. They are then going to be joined together to form a filament. Okay, so actin is going to be polypeptide made of these kidney-shaped G-actin, and the G stands for globular actin. Okay, the G-actin subunits, they have these sites on their heads, and let me just erase this. So if we look at kind of these sites right here, these are going to be where they're going to bind to the myosin heads. So myosin and actin are going to join and cause the actual contraction. The F-actin subunits link together, and then they form something called F-actin, which is fibrous, and so that would be then this kind of form right here. They then have 2 strands that kind of form this double helix around each other, or not necessarily like a DNA double helix, but a kind of a twist. Now, with the thin filaments, there are 2 other important structures. We have tropomyosin and we have troponin. And the tropomyosin and troponin are really important in the regulation of contractions. So what happens is tropomyosin is going to be this kind of thin filament. It's going to block all of the heads or places on the actin where the myosin heads could attach. Because if there's no tropomyosin, then these heads are going to be able to attach to the myosin heads, or sorry, these spots. Troponin is going to be kind of a regulatory agent that is going to be able to move the tropomyosin off of those binding sites. So when we talk about the actual contraction of a muscle, troponin and tropomyosin are going to be really important. Okay, so what's going to happen is troponin is going to bind calcium, which then is going to cause a conformational change in the structure. It's going to move tropomyosin off of those binding heads, or the actin subunits where the myosin binding site is, and it's going to allow myosin to actually bind there. We're going to talk about this idea of sliding filament model and talk about the actual how muscle contraction works, but I just kind of wanted to briefly give you a little preview of that. This image that I'm showing you right here is very similar to kind of how muscle contraction would work. You can think of the rope as being the actin, and the hands are kind of the myosin heads. And so what's going to happen is the myosin heads are going to attach to the actin and kind of pull on the actin so that muscles can contract. Okay, so the arms are kind of the myosin filaments, and the rope is going to be the actin. So with actin and myosin, there are a few other structure or proteins that are involved in this, and we're not going to go into too much depth about any of these, but just kind of mention them. Whenever we talk about certain physiological things, there's usually many proteins and many enzymes that we don't talk about, but these ones are ones you'd want to be a little more familiar with. So these are going to be proteins that help during this process. So we have this elastic element that is going to be called tintin, and I don't know if I think this is going to be, here's a tintin. So right here is tintin. So it's an elastic unit, and what it does is it allows for kind of holding the filament in place and helps with recoil. So you're going to see that there is an actual section that looks like a spring that's on the tintin. Another protein is going to be called dystrophin, and dystrophin is going to be in this bottom image. It is going to kind of hold muscle filaments in one place, and what they do is they attach to the muscle fiber membrane, so kind of the cell membrane, and the muscle filaments. And so they have a connection here so that you're not kind of having things floating around. Everything is going to be kind of stuck in place. So it links thin filaments to proteins of the sarcolemma. And then we have nebulin, myomesin, C proteins. Those are all going to help bind filaments of sarcomeres together. So you can see some of them, protein C there. I'm not sure if I could see any of the mesomesin, but those are going to be in that M line area. Okay, so again, these are going to maintain that alignment of the sarcomere. This is why when you look at across our histology of skeletal muscles, you see those dark and light bands because everything is aligned in the way it's supposed to be. Now, a little bit about, if we go talk about, we just talked about dystrophin, which is going to be involved in Duchenne muscular dystrophy, which is a common and pretty serious muscular issue. So what happens is the children have this, and it causes the muscles to basically just be destroyed, and it kind of continues on and causes lots of issues for the individuals. It usually is going to appear in early childhood, and it's a sex-linked recessive disease, so it's usually going to happen in males. It's less likely to happen in females because if it's on the X chromosome and a female has 2 copies of the alleles, they can have 1 bad copy and a good one and still be fine. But if they have 2 bad copies, obviously they will have Duchenne's muscular dystrophy. But it's rare to see this in women. So it's going to appear between 2 and 7 years old, and this is usually when the boy will become clumsy and start to fall frequently. Unfortunately, the disease progresses and it makes it kind of goes from the extremities upward, and it can reach the head, chest muscles, and cardiac muscles. And so if you're having issues with how the muscles are contracting, that's obviously going to be bad. These individuals can live, if they have supportive care, into their 30s and sometimes beyond, but it is something that is going to require lifelong attention. And what actually is happening is that there's a defective gene for the dystrophin. So again, remember, dystrophin is going to be this protein that attaches the thin filaments to the sarcolemma, or the cell membrane. And it allows for stabilization. If that's not functional, then these individuals are not going to have the stability inside of the muscle that needs to be there. So these individuals can tear their muscles easily, which then allows calcium to enter and damages the contractile fibers. There's then inflammation and regeneration, and overall it just causes kind of apoptosis of muscle cells. This is an image of a cross section. So this would be calf muscle, and so what you can see is that some of this has muscle fiber, muscle tissue, but what happens is when the muscles die, you have invasion of kind of just adipose tissue, so this is fat tissue, and obviously if you have fat that's replacing muscle, it's not going to be able to function in the way that it's supposed to. All right, so the last little structural thing we're going to talk about for today, and then we're going to, in the next lecture, start to move on to actual contraction. But we need to talk about the sarcoplasmic reticulum and the T-tubules, because these are going to play a huge role in how the actual muscle contraction is going to take place. So the the sarcoplasmic reticulum is going to be this blue structures, okay? And the blue is going to be kind of surrounding all of the muscle myofibrils, so kind of inside of the actual muscle cell. They're going to run longitudinally kind of down, and they're going to kind of be right on top of the sarcomeres. They're going to have these structures called terminal cisterns, which are going to kind of be at the very end. Let me just get rid of this. And so the terminal cisterns are going to be this area right here. So not the white, but the blue kind of overlapped the white. So you're going to have these terminal cisterns. So they're attached to the sarcolemma, or sorry, the sarcoplasmic reticulum, and are going to be kind of the end point that's going to then interact with something called the T-tubule. Okay. So the sarcoplasmic reticulum, its main function is to hold calcium. Calcium is essential for muscle contraction, and the sarcoplasmic reticulum is going to regulate kind of when calcium is released and when it's taken back up. Okay, so it's going to store and release calcium. The T-tubules are going to be kind of this white structure here. So I'm going to put the black on top of it. So this is going to be a T-tubule. So the T-tubules are going to be connected to the sarcolemma. So here, if we have this outside portion, all of this is going to be kind of the cell membrane or the sarcolemma, and this is going to be connected to the T-tubules. The T-tubules are going to go into the depth of the cell, so it's not going to stay just on the surface like most of the sarcolemma, but it's going to go deep into the actual muscle myofibrils. This is going to increase the surface area, and it's going to kind of continue. So this is technically connected to the extracellular space. What these are for is to transmit nerve impulses to the actual sarcoplasmic reticulum and the terminal cisterns so that they can release calcium. Okay, so the T-tubules penetrate, kind of go into the interior, and are going to interact with, in this area. So you have terminal cistern of the sarcoplasmic reticulum, you have the T-tubule. They're going to interact and are going to cause the actual neural impulse to create the kind of the right situation for calcium to be released, which then will cause the contraction. Okay, we call this area where you have both kind of the T-tubule, so it's called the triad, T-tubule and terminal cisterns of the sarcoplasmic reticulum. So this area is called the triad, and if you see, it has one, two, three structures, and so those three structures are going to be what we call the triad. Okay, so one terminal T-tubule and then two terminal cisterns are going to equal three kind of structures right next to each other. So the triads are going to be really important because they're going to be this this connection between the sarcoplasmic reticulum and where the nerve impulse is going to actually be coming in from the T-tubule. So the T-tubule is going to have kind of integral membrane proteins that are going to be on the surface of the T-tubule, and these are going to then interact with the sarcoplasmic reticulum. And so this right here, this is not from your book. I got this from an actual scientific article about muscle contractions. But you have the T-tubule right here, and this is going to be our sarcoplasmic reticulum. And so you're going to have interaction in this location right here, where you're going to have kind of passing of this nerve impulse that's going to allow for calcium to be released. So we will talk about this in much more detail, but there are going to be voltage sensors that are going to change shape if an electrical charge is coming in. Okay. The sarcoplasmic reticulum also is going to have integral proteins that will kind of protrude into the space, and they will then be able to kind of cause the opening of calcium channels. And so they'll have to be triggered to do that, and that triggering is going to come from the T-tubule. Okay, so when the electrical impulse is going to come, let me just erase some of this. So when the electrical stimulus is going to come, it's going to come down here, and then it's going to enter, and it's going to cause a change to happen in the actual shape of these integral proteins, which is then going to allow for calcium to be released. Calcium is going to leave and is going to start interacting with the actual myofilaments. Specifically, it's going to interact with troponin, which is going to be on the actin.